Hi, welcome everyone. Welcome back. We follow our talks with the second section of our talks. The first talk of the second session is Kisspeptin and Sexual Behavior by Dr. Marie Vedos. Dr. Vedos graduated as agronomist engineer in 2006 and obtained her master's degree in biology, applied animal production and health and agrocampus OS, France, to 2009. She carried out her PhD at the Caprine Reproduction Research Center at the Universidad Autónoma Agraria Antonio Narro in Torreón, Mexico, under a joint supervision agreement with the University Francois Rebelles in Tours, France. She obtained in 2013 the degree in doctorate in agricultural science and the degree of doctor in life science, both with honors. In 2015, she arrived at the Institute of Neurobiology at UNAM Campus Juriquilla for a postdoctoral position in the Laboratory of Sexual Behavior and Plasticity, where she worked on the role of opioids in male sexual behavior. In 2016, she was awarded a Council of Science and Technological Chair for Young Researchers in the same laboratory and remained in that position until she was hired at ENES Uriquilla. We welcome Dr. Marie Vedos. Thank you for joining us.
Hi, everyone. Thank you for having me today. I'm going to start sharing my screen, and please let me know if you can see it.
Now we can see it.
Okay.
Right.
So today I'm going to give you a little talk about kisspeptin and sexual behavior. And I'm going to start asking you, do you know why it is called kisspeptin? Well, first, it was Dr. Danny Welsh, who was working on breast cancer, who found that the KISS gene, because he called it the KISS gene, was able to suppress metastasis. And this wasn't because at this time, it was known that kisspeptin has something to do with reproduction, but because he was working in Pennsylvania State College of Medicine, which is in the state of Pennsylvania. And maybe some of you know that in this state is the city of Hershey, where Kisses, where the factory of Kisses is. And this is why he called this gene KISS1, right? After that, so this was in 1996. After that, all the family of kisspeptin peptides was described. So the biggest one is the kisspeptin 54, also called metastine for its activity. And there's other peptides. And the smallest one is the kisspeptin 10, which also have the same biological activity than the bigger one. And the interesting thing is that this peptide is common to different species, including humans, which only have one amino acid difference from the other species. And it wasn't before 2003 that kisspeptin was discovered to have a very important role in reproduction, with two articles that were published independently at the same time of different researchers that wanted to find the origin of lack of puberty or due to hypogonadotropic hypogonadism. And what they found is that these families had a mutation in the receptor of kisspeptin called GPR54, or also the kisspeptin receptor. And in this, the paper of Seminara, they also built a knockout for the receptor. And what they found is that in both males and females, the knockout had a lack of development of the reproductive tract and the gonads. And this was because there was a lack of kisspeptin signaling. So after that, there was a lot of investigation about kisspeptin and its role on generase secretion. Here I'm showing a sagittal slide of a rodent's brain, and in particular the hypothalamus and the hypophysis. And I'm just going to switch for the Montero. Sorry. so that you can see it there. So what we have is two main population of kisspeptin neurons in the hypothalamus. And in the arcuate nucleus, these neurons are located at the terminals of the GnRH neurons that you can see in green in this picture, with the soma located in the preoptic area, but they send the terminals until the median eminence where GnRH is secreted to stimulate the hypophysis. So in this place, kisspeptin neurons are now known as the generator, as a pulse generator, as they stimulate the pulsatility of the secretion of GnRH. And this is obviously very important for the pulsatility of the secretion of the gonadotrophins that will stimulate the gonads. And the other population is located next to the soma of the GnRH neurons in the preoptic area, in particular in rodents in the AVPV, the anteroventral periventricular nucleus, which is next to the third ventricle that you can see here in the picture. And in this region, it is sexually dimorphic, since in females there is more kisspeptin neurons than in males, and this is linked to the role of these neurons in this region, because they mediate the positive feedback of estrogens that will generate the LH surge necessary for the ovulation. So an important fact also that was discovered after is that kisspeptin neurons, in fact, possess or express the estrogen receptor, and this could explain how could be done the feedback of estrogens that are secreted from the gonads to the hypothalamus, right? Because before that, GnRH neuron doesn't express those receptors. So it was known that there was some kind of feedback mechanism, but it wasn't known where this feedback was actually done. So in this region, in the preoptic area, in the AVPV, kisspeptin neurons mediate the positive feedback of estrogen and progesterone to mediate, as I said earlier, the LH surge that is necessary for ovulation in females. And in the arcuate nucleus, in both sexes, it mediates the negative feedback that is necessary to maintain a good pulsatility of the secretion of GnRH and gonadotrophins that will stimulate during all the cycle the gonads, so testicle for spermatogenesis and ovulation in females. So kisspeptin neurons mediate estrogen feedbacks. But after that, there's also a lot more investigation that was done, and it came to a conclusion that really kisspeptin neurons integrate signals from the systemic signals or signals from the outside, as for example, photoperiod. There's in seasonal breeding animals, the seasonal breeding season is kisspeptin neurons are important to mediate the shutting down of the reproductive function or the axis during the non-reproductive season. And when the photoperiod is the good one, well, it's mediated by these neurons. The sex theories, as I told you right before, also kisspeptin neurons sense the sexual steroids, are receptive to sexual steroids, but also metabolism. It's known, the arcuate nucleus is a nucleus very important for hunger and nutrition and metabolism regulation. And now it is known that there's a communication between these nuclei, and that kisspeptin neurons are the intermediate between those nucleus and the reproductive axis. Also, something very important for sexual behavior is pheromones. In rodents, particularly, olfactory cues are very important to trigger and to stimulate motivation and sexual behavior. So it is now known that pheromones can modulate the activity of these neurons and hence modulate all the reproductive axis. So globally, all these investigation, basic investigation, but also investigation in humans have shown potential for kisspeptin to treatments, no? Like, for example, polycystic ovarian syndrome or precocious puberty. As I told you, kisspeptin is a very important actor in the onset of puberty, but also in other type of problems like pubertal delay. So you can see here is the LH pattern, which can be an increased pulsatility of LH or decreased pulsatility of LH, and treatment by kisspeptin are developing to try to treat that kind of diseases. But what this has to do with what I want to talk to you about today, which is kisspeptin and sexual behavior and reward system. Well, a lot of investigation has been done on fertility and kisspeptin, but now there's a little bit of evidence that show that kisspeptin can have an important role also in sexual behavior, which is quite not obvious, but important, because reproduction, I mean, sexual behavior is part of reproduction, right? So I'm showing here a picture where you can see on the left the different nuclei of the reward system, which is closely linked to the nuclei of this sexual behavior, because sexual behavior is a motivated behavior. So it evolves the reward system. And as I've shown you, well, we have a lot of kisspeptin neurons in the preoptic area. But it was also shown that there are kisspeptin neurons in the olfactory bulb and in the medial amygdala, which are also very important nuclei for the sexual behavior. And moreover, there are receptors in different regions of the brain, obviously in the medial preoptic area, but also in the ventral tegmental area and nucleus accumbens, which are two of the most important nuclei in the reward system. So, and also, sorry, in the medial amygdala, there are receptors for kisspeptin. So this evidence, and also the fact that there were a few studies showing that the infusion of kisspeptin in female and male rats can have an effect on the receptivity of female mice and on the erection, spontaneous erection without the presence of a female in male rats, well, have brought me to the conclusion that kisspeptin may play a functional role in mediating the rewarding properties of sexual behavior. So what was the question I first asked to myself is kisspeptin rewarding by itself? And for that, in rats, we use a paradigm called condition-place preference, which basically enabled us to know if a behavior or a drug causes the animal to feel good in a place, and we reinforce the behavior or the drug with being in one of the compartments. So we did that with 2 doses of kisspeptin, and what we found is that when kisspeptin is injected to the animal and we reinforce one of the boxes, we found that kisspeptin induces a very clear condition-place preference, which indicate that kisspeptin induces a positive affective state in the animal. We are now conducting other experiments blocking with an antagonist of kisspeptin and making the animal copulate to see if blocking kisspeptin will block also the positive affective state caused by sexual behavior. But let me talk a bit more about what is sexual behavior in rats. Well, in rats and in other species, there are 2 different and important phases. The first one is the appetitive phase, in which both sexes will investigate the conspecific other sex And in the case of the female, she will need to be in estrus in order to accept to be mounted by the male. So if she is receptive, which is totally dependent on hormones, on the estrogens and progesterone, then the male will start mounting. And this will cause lordosis, which is a reflex that I show you in this picture, which causes the dorsiflexion of the back of the female. And this is caused by the somatosensorial stimulation of the pelvis of the female, so that she presents the vagina and that the male can intromit. So eventually, after several intermissions, the male will ejaculate. And an important thing is that in the lab, since early studies of Dr. Paredes, who shown that when the animals are able to pace the sexual behavior, which means that they can choose when to go or not with the other sex, the sexual behavior will be rewarding. So it means that with the paradigm, the condition place preference I showed before, the animal will show a change of their preference. They show a positive affective state after copulating. So how do we do that in female? Well, we use this kind of box with a separation in the middle and a hole in which only female can pass through, because they are a little bit smaller than male, and we also train males not to pass through this hole. And like this, we can obtain that female pace the copulation, and it's rewarding for them. So what we do with this paradigm in these boxes, we put females that were treated either with a control cocktail of estrogen and progesterone, which we know that induces a good receptivity, as shown here by the lordosis intensity, because female can show different grades of lordosis intensity, which would be 2 the bigger one and 0 the smaller one. And it is known that both estrogen and progesterone are necessary to induce a total lordosis intensity. So as you can see, when we only injected estrogens, there was a significant decrease in the intensity of lordosis. But when we injected estrogen and kisspeptin, we had a significant increase, and actually even more important than in control animals, showing us that kisspeptin is responsible not to increase this receptivity. And when we injected both progesterone and kisspeptin, we also restored the lordosis intensity that we had with the control. Finally, what we did then was try and inject, it's behind the, I'm going to tell you, well, this is control, this is estrogens. And in this case, we injected kisspeptin with 2 doses of antagonists. And what we saw is that the antagonist partially reversed the effect of kisspeptin on receptivity, since we saw an intermediate level of receptivity in those animals that were injected with the antagonist. I'm sorry, that can be shown the legend. So finally, I only want to say like a resume of what I just told you, which is first that kisspeptin is an important regulator of the secretion of GnRH, LH, and ovulation. That kisspeptin acts as a sensor of signal that regulates the reproductive axis, and because of that, it has become the purpose of different research, clinical research, to try to resolve fertility problems in male and female. And part of my results that kisspeptin induces a positive affective state. And well, there's some research done in humans that show that kisspeptin is able to change the mood of the participant that were injected of kisspeptin. And finally, that kisspeptin increases the sexual receptivity in female. And well, thank you for your attention. I want to say thank you to the Instituto de Neurobiología, and particularly to Dr. Paredes and my student Edgar Ponce, who made these experiments, and to the founding the GAPA. Thank you.
Okay, thank you so much, Dr. Marie. We have a question. The question says, what does kisspeptin regulate naturally?
Sorry, I didn't hear.
The question is, what does kisspeptin regulate naturally?
Naturally, well, all the hypothalamus, hypophysis, gonad axis. Well, it's very important for the pulsatility of GnRH, so that gonadotrophins are secreted in a pulsatile manner, and that the gonads are stimulated. So naturally, it is present in the brain, and it stimulates the hypophysis.
Okay. Well, I think we don't have more questions. We want to thank you for participating in the Global Summer School. Many thanks for participating.
Thanks to you.
Okay, we follow to the final talk of our academic session. The following talk is named Astrocytes, the Start of the Brain by Dr. Mónica López Hidalgo. Dr. López Hidalgo earned her PhD in biomedical science in 2013 in UNAM, Mexico, understanding the role of astrocytes in synaptic plasticity and memory. After that, she conducted her postdoctoral research at the Max Planck Florida Institute for Neuroscience, where she worked on the role of astrocytes in visual processing in ferrets. She returned to Mexico in 2016 and the State University of Querétaro. Since 2017, she's an associate professor at Enes Juriquilla. Dr. Lopez has been a member of the National Research System since 2013. She was awarded the L'Oreal UNESCO CONACYT AMC in 2017 for Women in Science for her research on deterioration of cognitive functions related to age. She has many publications and presented her work in several national and international conferences. She's currently studying neuroglia interaction and its role in cognitive function and sensory processing in physiological and pathological conditions. We welcome again Dr. López Hidalgo. Thank you so much for participating.
Hi, thank you very much. I'll be starting sharing the screen.
Okay.
I cannot. So that I don't have the permission to do it. I think it's because Marie is still sharing.
Yeah, we have to see the. I think she needs to stop sharing.
Then I can share mine.
Go around. There is not a way I can.
Okay, maybe I can do some trick here.
Yeah.
Good, thank you. Okay, thanks very much for the introduction. It's really nice to be here and to share with all of you some of the work that we have been done in the last years. And so first, we have been for more than a year in pandemic. We still are not sure if we are sharing the correct screen. So is this the correct one? Are you seeing my screen? Good.
Yes.
Great. Okay, so my talk is about astrocytes, and I'm calling the stars of the brain. And then I hope that during these minutes I can convince you that astrocytes are actually the stars of the brain. But first, I would like to start with the most important thing is thanking the people that have been involved in all the work that I'm gonna show you today. So we have lab members. This is the current students. We have some former students. We have some colleagues from Enes, other from the Institute of Neurobiology, and also from the Max Planck Florida Institute, and of course to the funding that without money we can't do too much. Okay, so let's get this started. So what are astrocytes and why are we interested in astrocytes? And we call them the stars because it looks like they are stars, but now with more nice techniques, it doesn't actually look like stars, but before it looked. And when I say the brain, it's very likely that you are thinking something like that, right? This is a nice images with fMRI, and then you can see the tracts, and you can imagine maybe a human brain. And I mean, human brain is important and it's nice, and we have one, and we use our brain to study many other brains, but we can't forget that there are many other brains that are different, and that different species have evolved with different structures and organization that is just the best for them. And if I said brain, then you very likely thinking about neurons, because you know we have this neuronal theory that says that the brain, and in general, you know, the central nervous system, it's composed of neurons. And then you may be thinking something like this. This is, I don't know how to, wait. I can't go out of this mode. I'm sorry. Just need to do it again. Okay. Okay, so then if we see this neuron, this is a reconstruction of a neuron from a rat. This is layer 5. So this kind of work you can do when you feel a neuron with a dye, and then you can cut the brain and then see how is the structure of the neuron. You can see the dendrites and all the processes, and then you can try to understand how is the morphology of the neurons. And then we can analyze the function and the relationship between these neurons. And most of the things that we know about neuroscience is focused in neurons. It's neuroscience, right? But neurons are not alone. There are many other cells and just like many things that are composing this system that is the central nervous system. And it's not only about neurons. And we try to understand the brain, just understanding neurons, we are failing because it's just one part of brain. And we could just try to just put some other elements just to make this more interesting. And then we can see two very important stuff, that is the astrocytes. And astrocytes are very well located, that have a relationship with neurons and also have a relationship with the vessels. So, and even if we knew that astrocytes in general, like glial cells, are everywhere, and it was first thought that they only have like supporting functions like homeostasis, like ion balance and metabolism and neurotransmitter, regulated neurotransmitter levels. And these are very important functions for a system, because if you don't have an ion balance, or the metabolism is not well, or it's not regulating glutamate or GABA, of course you're going to have a pathology. Of course you're going to have a malfunction of the brain. And just based on the supporting functions of astrocytes, you can think that this is actually really, really important. But they're not only doing supporting functions, they are doing a lot more. But one question that we could ask is how many they are? Are they located everywhere? So if we see this image that I'm gonna try to put again my laser point, and I hope I can come back later. So, and if we see this is a ferret visual cortex, we have layer 1, 2, 6, and then we are staining neurons, and then we see the neurons in orange. Then if we would like to see how it look the astrocytes in these slides, then in green you will have astrocytes. So these green circles are the soma of the astrocyte, but all the processes are actually covering the entire gray matter. If we overlap both, it looks like that. If this has not already like surprised you, like, oh my gosh, they are everywhere, they must do something interesting, maybe this reconstruction can, yeah, maybe this 3D reconstruction where we can mark astrocytes and see them with a 3D reconstruction of 1 millimeter by 1.6 by 0.5. And you can see all the dots, all the blue dots are astrocytes, are real astrocytes in one piece of ferret visual cortex. So there are tons of astrocytes. They are regulating all kinds of things. And if astrocytes aren't working good, the brain is not going to work. It's not going to do its job. Okay, so what are the astrocytes doing? So some years ago, they found that astrocytes were surrounding many synapses. And it was called by Arak and colleagues in 1999, tripartite synapse, which means that it's not like presynaptic and postsynaptic. We have also astrocytes. And astrocytes have a lot of membrane receptors. We have ionotropic, metabotropic receptors. Astrocytes have substance that can release, that its name is a gliotransmitter and can act through modulating neurons pre and postsynaptic, and also establish this very interesting relationship with vessels. So they are now well known to be involved in synaptic transmission and synaptic plasticity. So let's go back a little bit about these different brains. So one thing that we asked a few years ago is if astrocytes are similar in all mammals. And then we compare two different species, mice astrocytes versus ferret astrocytes. So we wonder if they look the same, they're the same size, or if it was just a difference in layers or in species. So here we have mice, layer 2 and 3 from visual cortex. And you have, it's like a very nice, round, almost the same size. You can't actually distinguish layer 1 and layer 2, 3. But if you see ferret visual cortex astrocytes, then you have crazy astrocytes. It's just bigger. They have this stuff. It's just thicker. It's just like very complicated. And if you compare ferret from layer 2 and 3 from layer 1, it's crazy. It's like a walking astrocyte, right? That is have even, like layer 1 have like spine structures, like it's like really interesting to morphology. So then we compute the volume of these astrocytes. So we have the ferrets are almost twice bigger than their counterparts in mice. So something that it's a rule that you can even see, in this very famous book like Candel. It's about the non-overlapping domains. That means that astrocytes have a territory and they just touch like just a little bit. And so all the gray matter is covered by these astrocytes. They are sharing just a little bit, and they do a lot of stuff. They're regulating how neurons function there. And then so we wonder if this rule applies also for all the animals, or is just rules for rodents. So then we fill the astrocytes. These astrocytes were filled in vivo. So we went into an animal, we filled the astrocytes with a dye, and then we took the image with a 2-photon microscopy, and we reconstructed. And then we saw that, yes, you have the yellow one and the purple one, and they are actually, not really overlapping a lot. So you can see like this is rotating the astrocytes, so you can see from different angles. And yes, they are not touching too much. There's just a little bit that they're touching. So the rule about non-overlapping domains is true in mice. How is in ferrets? So then we have here 8 astrocytes that were filled with different dyes that we could see how they relate each other. And then this silver stuff that just appeared, it's just how much they are sharing. As you see, it's a lot. So you can see like some cuts. There's some astrocytes that just share a little bit, but some others share a lot. They're really intermingled. So we know now that ferret astrocytes doesn't have these non-overlapping domains. It really depends on the distance between these two astrocytes. So the further you are, the less you're gonna overlap with your partner. But then we found a population that doesn't follow the rule, the ferrets rule. And it's just this one that they are the closest, but they don't share almost anything. So then we look at them, and then we found these guys. This is the kissing astrocytes. So they kiss because they are they are touching, but they don't hug, right? So then the process going to one side to the other side, but they don't actually touch. And okay, so astrocytes are different. They do a lot of things. They do supporting, they do regulate synaptic transmission and plasticity. And we knew all this because all the experiments that it was done in lysis, in vivo. But something that took our attention into glia and into astrocytes was that we saw that they were not silent, that we previously thought. Because when you do electrophysiology and you have a neuron, you can see spikes or you can hear it. But if you have an astrocyte, then it's just silent. And the people say, it's just silent, it's not saying anything, it's not talking to neurons, it's just quite mute. And it wasn't the truth. It's just because we were not seeing them, because they don't produce action potential, but they actually have a lot of activity. And then here I'm showing you some calcium activity, spontaneous activity. So that means that ferret astrocytes are just active. Even if the ferret is with their eyes closed, you have this kind of activity. So that you can see in yellow, it's more activity, which means more calcium. So we have like spikes, calcium spikes from the astrocytes. And okay, so they are not silent. They do a lot of stuff. And we know that they are not doing only supporting stuff. So a few years ago, we were wondering how they were involved in synaptic plasticity. So we work on lysis in a hippocampus. We work in this synapses that is very simple to work with synaptic plasticity. We were working on the Schaffer collateral stimulating, and we were recording in CA1. And we have this. So we know that in this circuit we have astrocytes, right? So at that moment, there wasn't so many techniques to silence them or activate them. So we used what there was before, and there was a gliotoxin that is fluorocytide that is just like decrease their activity. And so we have here an LTP that is the base for long-term memory. And in the presence of fluorocytide, you can see that there's no LTP, which means that LTP requires the activity of fluorocytide. And we know that this LTP depends off NMDA receptors and are very important for plasticity. So we wonder if astrocyte regulating NMDA receptor synaptic plasticity dependent. So we target viscerin because viscerin is released by astrocytes and it's a co-agonist of this NMDA receptor. So what we observe is that if we supplement viscerin, then we are able to fully restore these LTP. That means that astrocytes are important, and not only astrocytes, it's just astrocytes through the release of viscerin. So then we wonder if LTP is involved, if viscerin is involved in LTP, if it was involved in learning and memory. And yes, if we block astrocytes, they have a decrease in the long-term memory, and we totally rescue when we supply viscerin. So finally, we ask, like, if this is only young animals, but what about aging? Because cognitive decline associated with aging depends also on NMDA receptor. Is viscerin that comes from astrocytes also important? And so what we did is just a task for cognitive flexibility. Is when the more perseverations that you have to do something, the less you have of cognitive flexibility. And I'm not going to explain a lot of this task. So I'm just going to say that we the rats learn that they need to press a lever when they see a light, and then they receive a reward. Then they learn to do it like really well. But then one day we changed the rule. So they see the light, they press the lever, but they don't actually receive the reward. So the rats just became crazy, like they press lever. And we count these pressings of the lever without receiving rewards, like perseveration, like it's the inverse of cognitive flexibility. So what we have is that we have young animals, middle-aged, and aged animals. And of course, we have an increase in the perseverative errors. That means with age, as we aged, we have less cognitive flexibility. And so we wonder if discerning was involved in this. So we try a higher dose of discerning, and we were able to reverse this cognitive decline, which are with the lower doses, and it was not effective. So this is dependent of the doses of discerning, which means that discerning reverse cognitive flexibility decline associated with age. So we're working on this. We have a lab that's working with neuroglia interactions. We have this aging project. We have also, I'm now showing you this information about sensory processes and cognitive function. And we combine behavior, electrophysiology, calcium imaging, chemogenetics, optogenetics, and many other stuff, and many things.
Many thanks, Dr. Lopez Hidalgo. We have time for one question. The question is there a neurological disorder that can be attributed to glial dysfunction?
There's many disorders that can be attributed to glial disorder. Like, I don't know, there's so many that I can't even start.
If you can give us like more representative, like.
I know, sclerosis, it's an autoimmune disease that affects the myelin. The myelin, it's made by oligodendrocytes and Schwann cells that are glial cells. I think this is like the most typical glial pathology.
Okay, thank you so much again for your participation in the Global Summer School. Before we go to our, well, with this, we finish our academic sessions. Before ending wrapping up the day, we have our last cultural session, but before that, we will, Dr. Magda Giordano will give you some information about Enes Juriquilla. Thank you so much.
Thank you. We are setting up the last part of this day's session. And so I'm gonna, well, we set up everything to show you what would it look like if you were here in Juriquilla and you were finishing up your your meeting sessions. Where would you go and what would you eat? So while we do that, I'm going to explain a little bit to you about what Juriquilla is. And see, so we're almost ready to go. So Juriquilla is part of Querétaro, as Mariana told you. And we, which are the Enes Juriquilla, are part of the larger university, which is called the National University of Mexico, UNAM. So we are in a different, the main campus of UNAM is in Mexico City. It's a beautiful campus that you have to visit someday with wonderful and interesting buildings and mosaics. And from there, that's where the main campus is. But at one point, the university decided that it had to have campus outside of Mexico City to be really a national university. And that's how we came to be. And it was in around 1996 that the campus in Juriquilla started. And first with the Instituto de Neurobiología, and then other centers of in research in physics and in geosciences, and the laboratory of the genome that Maria Avila belongs to, and then the Enes, which is also the Institute of Mathematics. So we have several centers and institutes that do research in the campus, but we belong to the National University, although we're not located in the main campus in Mexico City. And as Mariana described, we are in Querétaro, which is ready, which is in the center of the country. So we're ready, and I'm gonna give the voice to Monica, who's gonna continue with the last cultural session of the day.
Hello, everyone. So this is the last session. This is a cultural session. So before I talk about astrocytes that are actually really cool, but now we are going to talk about some other stuff that it's not that cool as astrocyte, but it's kind of similar. Okay, so we're talking about something that we really like in Mexico, you know, because we're talking about a conference dinner that you will have if you come to Querétaro. And one thing that you have to know is that at UNAM, we don't usually host like dinners inside. I don't know if it's because alcohol is not welcome or, you know, like, I don't know, but it's not like usually we have dinner inside. So usually you go outside and then you can eat a lot of things. But if you come for a summer school, even if we get you to some place, you are gonna meet some students and they are gonna take you to eat something related with Querétaro or Mexico. So let's travel a little bit about what are our roots and why we eat what we eat. So first, I'm going to talk about something that is eaten by our students. So this is our real pictures of them. And we like corn. And maybe you're thinking corn, you're thinking this kind of image like with butter, and it's the yellow one. But maybe here you wouldn't find this like very often, but then you'll have this kind of corn that is toasted, and then you can put like lemon, it's not lime, it's the green one, and salt. And then you can, here in Querétaro it's very common. So if you're driving to go to the missions or to go to some small town in the street, in the highway, you're gonna find like small places where you can get this kind of corn. And this also is very common. I think it's like maybe around the country. It's just the corn with mayonnaise and cheese, and of course, chili. But actually, this is more representative of what we have in this part of Mexico. So we have corns of many colors, not because we tinted, it's because they are like that. We have many varieties. And then, of course, our produce comes in different colors. Sometimes when I was outside, people ask me, like, how did you paint your corn, your tortillas? And I'm like, no, we don't tint it. They are just like, the corn is that color, right? And it's just, look at them, it's just beautiful colors, right? So then we prepare tortillas, and the good tortillas must like inflate a little bit. You know, this is a good tortilla. And also we have tortillas that are like this color, the color of our corn. And then we have quesadillas. The quesadilla is something like, it's really delicious. I love quesadillas. I grew up eating quesadillas like all day, all night. And we have kind of a debate between Mexico City and the rest of the country about quesadillas. And maybe our fellows from Mexico City, when it's their turn, they can explain us why they think that the quesadilla is without queso. But the rest of the countries think that quesadilla comes with queso and many other stuff. This is a debate in Mexico, and there's tons of memes about this. And here in Querétaro, you can go to the market that Mariana told you in the morning, and then you can have this, you know, my mouth is watering. And then you have these gorditas. Gorditas is something very typical from Querétaro. And then you can have like cheese, or you can have a lot of things. And sometimes we put like lettuce, because we don't eat a lot of veggies, but then because we try to be more healthy, we put lettuce. And it's really delicious. You can toast it or fry it, and it's really good. And now let's talk about tacos. And then maybe you have seen these kind of tacos, or you have eaten. And the question is, are these Mexican tacos, real tacos? And no. Just to make it short, no. These are Tex-Mex tacos, and I'm not saying it's bad. I never tried, but maybe they're just delicious. It's just that they are not like local from here. It's like from pizza. Maybe if you are in Italy, and then what I'm gonna say, maybe you are gonna feel like disappointed, but we put pineapple to the pizza, and then you're saying like, oh my gosh, how is that possible? Well, I mean, different countries, we do like many different stuff. So this is Tex-Mex tacos. I'm not saying this is bad. I mean, they are really delicious. It's just this is not Mexican tacos, right? And it's something weird because the taco is stand by itself. It's like holding. And they have these like taco holders. I don't know why people want the taco hold by itself. It's like, doesn't make any sense, but they have. We don't have anything like this in Mexico. And then there's some advertisement, like you have to take the taco like this. No, please don't. And maybe this is more like, it was a copy of this kind of taco that we have here, like it's a fry, tacos dorados, another place called like flautas. And, but now we're gonna talk about soft tacos. And then, so soft tacos can be eaten in the morning, in lunch, in dinner, like 3 in the morning when you're out of the bar. And okay, so let's talk about these tacos. Okay, so today we bring some of the tacos that we have here. Look at this. Let me see if this adjusted. So it's not adjusting the camera as usual. Okay, so this is hot mix with tortilla, of course. And so we have pastor, chorizo, chuleta, carne asada, and something that you have to know that is really important, just let me read one of my tortillas, is that the tortilla has a side. So there is the thin layer and there's the thick. The meat must be in the thin layer, okay? So we have many salsa from different colors, different chilies. We put some veggies, you know, we like veggies. So onion and coriander and lemon. But I can tell you a lot of things about tacos, but it's gonna be boring. So why don't we invite a person that just talked to you about Kispetin. She's a researcher, she's a professor, and she's French, and she lives here in Mexico. So it's maybe nice if she said something about how is her experience in tacos.
Well, I've been in Mexico for more than years.
The camera is losing us.
I've been in Mexico for like 11 years, and at the beginning it was a bit bizarre to me, tacos, because I'm French. So for a start, I'm not used to eat with my hands. We usually are. We have like very special manners. And then the salsa, yeah, because the most spicy I have is pepper, and here with all the chili they have. Well, there's some stuff that is really, really hot, and if you're not used to it, you will not enjoy the food. And then you have the Mexican people that are very used to eat spicy food. So if they say to you, no pica, which means it's not spicy, it means, yeah, it's spicy. And if they say it's spicy, it means that it's really, really spicy. So I'm sure maybe you've seen the meme, because it's very funny fact about Mexican people. And that's very true. I mean, I think every foreign people coming to Mexico, the first time you have a taco and say, no, you have to put in salsa because the taco with salsa is not a taco. Well, yeah, but you have to get used to it. And some people not even get used to it. Now, I agree that the taco must have salsa to be good, to be a whole taco. And also, I totally agree with the Tex-Mex, because in France, it's exactly the same. I mean, they sell this kind of, which would be maybe a tostada, which is a toasted tortilla, but it's not Mexican tacos at all.
Yes, so if you are living outside of Mexico, maybe you want to share your story about tacos in your hometown, and then you can write it on Facebook or YouTube and share your stories with us. Well, we continue talking with our French-Mexican researcher. So, Marie, what is your favorite taco? Which one?
I would say pastor. I really like also carne asada. I don't know how to say that in English, but I think it would be pastor. But the best one I've had is in Mexico City. It's actually one of these local industries that Monica told you about, and they were spectacular. I mean, these guys called Los Cuñados were the best tacos I had in my life. But yeah, you can find very good tacos in every city. They will have their own specialty. For example, I've lived in North Mexico before, so I knew very well the gorditas, which is another kind of food made with the corn paste, but not in form of tortilla. But it's filled with meat and all kind of food, and it's very good.
Yeah, it's true. I mean, the more delicious tacos are usually in the street. But yes, as Mariana said in the morning, you can get sick. Have you get sick?
Yeah, at the beginning, my stomach had to get used to it. Now, I think I'm okay. Sometimes, yeah, maybe if I go to a place that I don't really know, or maybe because it's too greasy, yeah, but globally I'm okay, and I love Mexican food. I mean, I've learned to cook some stuff. I love cooking, and the first thing I cook is rajas, which is another thing that is very special about Mexico. It's with special pepper, which is from Puebla. It's a big pepper. It's not that spicy, well, it can be spicy, and it's with cream.
It's not spicy.
Sorry. Thanks.
Okay.
Okay.
I think it looks really delicious. It is breakfast time in Mexico. It's dinner time in Europe. So you can grab a taco. Find some taco stores. So now, I'm sorry for the people in Europe, but I'm pretty sure they will find a place where they have proper tacos. Probably not as good as these ones, but with this, we want to conclude the session of today. Thanks so much to everyone. Thank you so much, Dr. Lopez Hidalgo, for your talk and for your gastronomic experience. And before we want to invite you tomorrow, we follow with our Global Summer School 2021, day 3. Before we conclude, Dr. Araida might want to say a word. Thank you so much to everyone, and we'll see you tomorrow.
Yeah, do not go yet, please, because we still have that feedback that we are trying to get after every session. So thank you. Yeah, it's very cruel to show all those delicious tacos to us that we cannot get the same quality. So thank you very much. When, as I say, text and send some messages, I cannot wait to visit you all in Juriquilla. And of course, in addition to discuss scientific topics and plan collaborations, and maybe bring students and organize joint events, of course, to have a good time together. So thank you very much. It's been a very informative, thought-provoking scientific talks. So interesting. I'm sure there are lots of opportunities for collaboration as well. But before we go, we want you to help us with our VBox feedback session. And you know how it works. You know how you either get your smartphone, your tablet, another device, or just open another tab. And if you could go into vbox.app again and just type the same number as yesterday. So 178-384-041. And I'm hoping we're going to get even more respondents than yesterday. Although, just in case you are not aware, I do not think we will have many British students today because England is playing Germany in the Euro, and it started 25 minutes ago. So I'm afraid they might have missed the tacos because they are watching their team play. So, but without further ado, let's get on with this one. Do you know what is the what you have to answer. So again, which institutions are you from? We only have 10 people registered. Come on, guys. Take your app, take your smartphone and just go into bbox.app. It cannot be that yesterday we had 36. Don't tell me that those 26 missing are British watching the football. Come on. Do register, bbox.app with those 9 numbers. And where are you from? Are you from Enes, Opibi, Novara, or Manchester, or any other if we have additional? And I'm going to close in 5, 4, 3, 2, 1. Okay. And we can see that, oh, we still got some from Manchester. Well done. Well done. I'll be very quick so we can go and watch the rest of the first half. Thank you very much. So which country are you from? And again, just type the word. Where are you from? Where are you watching us from? Just especially for those that said other, we want to know where you are. Are you in Mexico? Are you in the UK, Italy, Germany, Japan, China? Where are you? Where are you right now? We got another respondent. Perfect. 13. Yay. We're having more. It's recovering. Remember to go. It's the same that you did yesterday. Exactly the same, because we want to gather all the responses and have them together so we can compare and see if whether you all thought the same. So 5 seconds, 4, 3, 2, 1, and closing. And where are you from? And it's going to tell us Mexico mainly. Oh, one from Nottingham. Excellent. Nottingham University. Good. Next one is, which field defines your course best? Are you from a medicine background, biomedicine, biotechnology, bioengineering, neuroscience, genetics, orthosis and reconstructive, or any other? And from the 13 respondents, that's a quick one, especially if you were with us yesterday, you pretty much defined which is your field by now. So which one is your course best aligned? Come on, guys. We only 50% responded so far. Just a quick click. Yay, we got 15. It's increasing. So excellent. I'm gonna close. We've got everybody responding, and oh, we can see other. That's very interesting. That's different from yesterday, definitely. So that's gonna be interesting. Maybe pharmacy. We should include pharmacy in there. Okay, just remember, all the answers are anonymous. We're not matching responses or cross-relating, so we do not know who you are. So please let us know which is the gender that you best identify to, so we know who is engaging the most. Is it female students, male students, or we don't know what type of students we have. Let's see if there's any gender balance. I think today was pretty good, actually, good balance of speakers and presenters. Yay, we got 17, it's increasing. That's really exciting. So from the 17, I'm going to close in 5, 4, 3, 2, 1, and closing the poll. And yay, we still got more female. Yay, girl power. But now we are going to hopefully, maybe we just have lots of male students just not bothering replying to these questions. Do engage, please, because we do want to know the feedback. So which session did you enjoy the most? Cultural sessions, scientific sessions, or both equally? What do you rather have, just a scientific summer school or just a cultural summer school? Or are you enjoying that we are mixing both and having some cultural and some scientific? And it seems like we lost one respondent. We had 17, we're down to 16. Yay, we recovered that 17. 5, 4, 3, 2, 1. Oh, it's everybody answer. Okay, almost 3/4 of you are enjoying having both, and then it's a bit divided on the other one. So that's a question that is going to be a bit different from yesterday. Well, it's the same question, but obviously yesterday you all knew a bit of English. So if you're not from Mexico, which is today's hosting country, are you more inclined to travel to Mexico in the future after COVID, when everything is allowed and restrictions relaxed? Would you travel to Juriquilla for an event, course, conference, or just on holidays for any reason? It doesn't have to be business related. It could be just for leisure. So would you go to Juriquilla after learning all that about those magic towns, beautiful, beautiful and so colorful. So what do you say? We're going to give 5 more seconds. Yes or no? 5, 4, 3, 2, 1. And wow, everybody's convinced that we have to go to Juriquilla. So definitely we need to get planning something for when it's allowed. Next one is, if you're not from Mexico, are you more inclined to start learning Spanish if you are not fluent or is not it was not in your priorities? Do you think now that you need to start learning Spanish, especially if you're planning to come to Juriquilla? What do you think? Yes or no? We've got 18 respondents. Excellent. And this is a quick one. Yes or no, not many options. I'm going to close in 5, 4, 3, 2. I want to wait a little bit more. Come on, we're missing 5 respondents there. Another 2, like we say in Mexico, there's another 2 and 1. Okay. So, yes, most of you want to learn Spanish after today, and some, maybe the other one already knew Spanish. Okay, next one. Would you be interested in participating in other international summer schools? And that's similar to yesterday. Do you only want face-to-face? Do you only want virtual? Are you not bothered and either way is fine? Or you say, no, I've had enough of summer schools and that's not what I need. So we've got 10 responses already. I'm gonna be 5 more seconds. 4, 3, 2, 1, and closing. And what's it? Face-to-face. I can see it's quite divided. It could be either or face-to-face, but yeah, we do miss people. We do miss human interaction. So what did you enjoy the most from today? Remember, responses will not show. This is the one that you can actually type. And we are not going to show here whatever you're typing, but it's going to help us to keep planning and improving summer schools and courses, virtual events, as well as collaborations among these institutions. So let us know what you really enjoy, what you want to see more of something. If you want to see more cultural sessions, maybe instead of 2, you want 3. Maybe you want a dance lesson instead of a tacos lesson, or in addition to tacos lesson. So let us know what you enjoy, what you think we should repeat, or not exactly repeat, but do more of that. Equally, if there's anything that you said that you thought maybe this could be improved by doing something, let us know as well, so we can do that, and we can take on board all the feedback and comments that you share with us. Again, this is not shared on the screen, so nobody but the organizers will have a look. And we will not take it personal. We want to improve. We want to make sure that you are making the most out of this summer school. So thank you for all your comments. I know this takes a little bit longer because you have to type. Hopefully, again, now because you know how this works, is easier. And I'm going to give 10 more seconds. No pressure, but I know that especially those who are in Nottingham in the UK want to go to the football. So 10, 9, 8, 7, 6, 5, 4. Oh, I should be counting in Spanish. Yeah, I forget this. I'll get it for tomorrow with Italy, hopefully. So we are closing now responses. And we have the data capture. That's great. Thank you very much for joining us today. I'm sure that you appreciate all the effort that organizers and speakers are doing to make sure that we host a great event for you and you make the most of it. And please join us tomorrow. We have a very exciting day from Italy, from UPO Novara, and I'm sure you will enjoy the session as much as you have enjoyed these 2 days. Thank you very much. See you tomorrow.